the old way we did things can be costly and painful.
Current detection technologies, however, are very limited. Everyone has the right to use these technologies.
It's really thrilling how we make use of the latest in material science and the whole world of nanotech.
This would not be possible without everyone's contribution.
Good afternoon, Dr. Xiaohuilin. We are so privileged to have you here today.
Thanks for having me.
Yeah. You know, to kick off the whole thing, we are really very grateful you come by because your background reads like a long list from physics to biology to biomedical engineering to your pet topic on nanotechnology. Can you tell us why you went into it and what is so thrilling about this whole area, because it's really out there for a lot of people? Maybe you can explain to us a little bit about what you're doing and why you're doing it.
I've been very interested in physics and biology since I was young. And it's very nice to see that they can be integrated to address real-world biomedical challenges, particularly in the area of nanotechnology. So a lot of molecules that are very useful to indicate whether we are healthy or are we actually facing a challenge. They are actually very tiny. So what we are doing in laboratory is to build very tiny sensors that match in scale to these biomolecules in our body, such that we can review novel and reflective information about our body to help with very early as well as safer detection of diseases. I would like to use the example of a recent technology that we have developed, which is called the APEX technology. It's a technology that we actually developed such that we can now monitor and detect Alzheimer's disease from a drop of blood.
Maybe you can tell us what's the difference in these biomarkers. What exactly are they?
There are many different types of biomarkers that we can find in our body. Traditionally, when we talk about some of these biomarkers, let's say, for example, in the case of oncology cancer, one of the most direct way to extract molecular information about the disease is actually by doing a tissue biopsy, let's say for a solid tumor, so that we can obtain tiny pieces of these tumor tissue and then analyze them with molecular technologies or under the microscope to look at what kind of nature they have. There are other types of biomarkers that we are particularly interested in, and these are actually circulating biomarkers. What that means is that they are actually biomarkers that can be found in easily accessible bodily fluids. That means blood samples, urine samples, saliva, fluids that we can actually obtain quite easily without subjecting the patient to go through this type of invasive biopsy process. And the key advantage is that means we can actually obtain the sample easier. It's also possible for us to do a serial monitoring, which means that we can actually longitudinally observe what type of changes are happening during disease progression as well as during treatment. Then we can evaluate better whether patients are responding to these treatments or not in real time.
So we back up. Say for oncology, normally we'll use markers like CEA, prostate, PSA.
Yes.
In this case, you're taking markers that are far earlier in the disease. Is that what you're saying?
Yes, which are markers that are directly secreted by the tumor. A lot of the conventional biomarkers that we are detecting, they might not be so specific. They're representing a very general idea of the disease. But novel biomarkers that we are looking at in circulation, these could be things that are directly released by the tumor themselves. They can present ideas about whether the tumor is actively growing. Are they responding to treatment or are they dying? So these are very, very specific biomarkers.
So they're like proteins, RNA, even ribosomal content. Is that in that range? That's a lot earlier?
Yeah, and there are actually quite a big spectrum of these circulating biomarkers. They can have different sizes.
Yes.
We talk about the size dimension, that's really important. These can be bigger materials such as cells, which can be cancer cells that are just liberated from the solid tumor.
Yes.
They can also be small lipid bubbles, like really tiny lipid bubbles that are released by tumors when they're actively growing. There are also metabolites, free-floating proteins, and nucleic acids that can just be released at different stages of the tumor progression.
That's actually amazing. And what do you actually do? You deal with nanoparticles that actually are impregnated with the equivalent matching reagent? What exactly do you all do?
Yeah, so these biomarkers have a wide variety of different sizes. So we try to use sensors, meaning detectors that match in terms of size and dimensions to these biomarkers. And we can explore two strategies. One is using metallic material, solid state material, where we pattern, impregnate them with different features that are matching in terms of size, such that we can use physics principle to look at these biomarkers. We can also build molecular sensors, which are using what we found in nature, in cells, like nucleic acids, proteins, and enzymes into molecular machines that can inform about these biomarkers. So across this spectrum of circulating biomarkers, we will design ways that we can interrogate specific components of these circulating biomarkers.
Fascinating. That's fascinating because I think to bring the audience back, why is this important? Because the old way we did things, it can be costly and painful. For example, when you have to actually cut out a tumor or do a biopsy, whatever it is, there's a certain amount of discomfort, morbidity, even mortality. And second thing is, it's difficult to do because in many places there isn't even a hospital that can do it or the specialist can do it. And now the day and age of everything from material science to information technology, we can actually combine all this and basically come out with kits that can actually test a whole range of diseases.
Yeah,
you know?
absolutely.
Okay, so we've talked about cancer. Maybe we go back to Alzheimer's. Tell us what exactly, okay, the difference again between what's conventionally used to diagnose a person with Alzheimer's versus what you can offer in this world of nanotechnology.
Okay, Alzheimer's disease is the most severe and common form of dementia. Current detection technologies, however, are very limited in detecting this disease, and therefore currently it's always detected at a very advanced stage. And also, without a good diagnostic platform, it's very difficult to also develop drugs that can address some of these molecular changes and finding a way to cure this disease. And what we are trying to do is that we are developing a blood test that can really look at these very unique changes in the brain at a very early time point to look for these very sticky biomarkers that cause the damages in the brain.
So you're looking at stuff like the beta amyloid?
Yes, the beta amyloid.
But my question is, normally the amounts found in the blood are very low, right? How do you all detect it from the blood?
Yeah. So some challenges of looking at this beta amyloid is that first of all, it occurs at very low concentration, very tiny amount, trace amount in the blood. And the second thing is that traditionally, if we were to measure just the beta amyloid, this biomarker, and we compare it against, let's say, brain imaging changes, there isn't any correlation or very poor correlation. Which means that if I were to do a blood test, it's as good as saying throwing a coin to say whether it's reflecting the brain changes or not. And the way that we overcome these challenges is by developing these nanosensors that are very sensitive, over 10,000-fold more sensitive than conventional technology to look at these circulating beta amyloid proteins in the blood. And the second way is that we are using this technology to really tease out the most reflective and the damaging population from these protein markers. The very sticky population that will grow into bigger patches to kill the brain cells and therefore causing symptoms.
I mean, honestly, you could extrapolate that to almost every disease, right?
Yeah, so that's the hope that, with all of these novel and advanced technology, we'll be able to address a lot of these different diseases, addressing the unmet needs.
What are the ones that are readily available in the market right now?
So there are a few technologies that we are actually trying to commercialize and bringing it closer to patient use. One of which related to the current situation is actually this COVID-19 test that we are developing. And the hope is that with the global pandemic situation of COVID-19, that we are able to bring a lot of these molecular tests for COVID-19 out of the laboratories nearer to the patient level, such as in the doctor's office.
On Alzheimer's, is that ready, or any of the other diseases?
Yeah, so Alzheimer's disease is the one that we actually developed to validate the technology. Currently, we are discussing about what are the ways that we are going to commercialize the technology. And our hope is always that we will be able to bring the technology to the market such that more patients can actually benefit from this type of novel technologies.
Where would it stand in terms of cost?
Yeah, it's a very good question. And one of the key challenges about this type of imaging, particularly for Alzheimer's disease, is that it's very, very costly.
Yes.
In Singapore, let's say for a single scan, it will cost about 2,000 to 3,000 Singapore dollars. So it's very difficult for patients to go through multiple scans, for drug development tests to happen, which means that they need to recruit a lot of patients, and every single patient will have to go through this kind of scan. And with this type of nanotechnology that we're building, the cost can be less than 1% of the current cost of brain imaging.
Wow.
Yeah, so that will be something that I think will be able to move not only the research field forward, but also definitely benefit a lot of patients.
That's really good to hear.
Yeah.
Are there many that are actually in this direction globally, or it's just a developing field?
It's a rapidly developing field, and we also see that there are a lot of opportunities for actually bringing this to market to commercialize.
Yes, and even for collaborations worldwide,
exactly,
right?
Yeah.
Because I would imagine one center will want to focus on metabolic disease, another center on oncology.
Yes, yeah.
So I assume a lot of that's happening, right? The interconnections and the collaborations.
Right. So a lot of these technologies are actually what we consider as platform technologies, which means that we show a concept and we show that it's working very well for one indication, but beyond that, it can be expanded to a lot of other diseases. So with basically this group of global collaborators that we have, we're able to explore a lot of different indications, disease aspects, as well as ways that we can further improve the technology.
Would you like to share about your dream for all these platforms? I guess that's the nice way of putting it.
From these novel technologies, there are two areas that we would really like to focus on. Primarily, the first area is about how do we exploit and develop complementary technologies that we are able to empower new discoveries in biology, to make these biomarkers talk, to reveal more information about diseases that previously we were not aware about. Another area that we would like to embark on is actually making these technologies very affordable so that we can bring these very innovative, sophisticated technology really close to the patient level. Our belief is that everyone has the right to use these technologies to actually really help with the public health and make this a better place.
Thank you so much for coming by. Really appreciate it.
Thank you very much.
Yeah.